So I talk about computer tools for paleoneurology, and you see that Simon is an author too. And basically this is more of a companion talk to Simon's talk. So the really interesting stuff you'll hear tomorrow from Simon. I'm only going to talk about the petty methodological details, but of course sometimes these details, they do matter, and I'll try to show you why I think that it needs to be anal about something. So again, I really want to thank the organizers, and I want to thank a lot of people for providing access to fossil material and to extant specimens, and being open for discussion and collaboration that would not be possible without a lot of other people. So I talk about three main things today. I talk about homology, which is a major issue when we deal with brains in general and endocasts in particular. And I briefly want to talk about asymmetry, brain asymmetry, or endocrine asymmetry. I think I definitely want to learn more about what AIDA is doing right now, because I think we're doing very similar things. I mean, from the quick flashes of the presentation that we did not get to see. And maybe the biggest part of my presentation will be devoted to a topic that we've been dealing with for quite some time. The problems that come with uncertainty of the fossil record. And we have uncertainty that derives from having very small samples, and we have uncertainty that derives from fragmentary specimens. And I want to show you how that impacts our ability to say something meaningful about human evolution. And I want to show you examples and ways of how we think we can cope with seemingly insurmountable. What I will not talk about is how to create an endocast. I think this problem has been solved, and there are many ways to do this. All of them are decent, I would say. You can do this automatically if you have a specimen, a recent specimen without matrix inside. You have to do this time-consuming and labor-intensively if you have a matrix-free fossil specimen. But basically, all these answers, they do converge on all these different methods, they converge on similar answers. And so I think that has been solved years ago, and what made you the author of a science paper that made it on the cover of Science 20 years ago, that's what our trainees learn in the first week when they start in our group now, so I think this is solved. Also, what we now know is how we can measure size of an endocasting. That's an easy computational problem. What's not so easy is how we can compare and particularly quantify shape differences. I think we leave it to tomorrow's talk and also to the discussion what, if anything, endocrine shape really is. But if it is something, then we should be able to quantify it properly, and that's the main focus of my talk today. So just to get the, I would say, preliminaries out of the way, so when I talk about shape, I mean it in the geometric morphometric sense or in the mathematical sense. I'm talking about the properties of an organism that are independent of location, rotation, and scale. One of the, I would say, techniques that have been very popular over the last decade at least is a set of tools called geometric morphometrics, and here you can see points measured on a skull, or actually on two skulls, and then they're being superimposed and they're being scaled, and what remains is the shape information. So that is something that we deal with for the rest of my talk. So let's talk about homology first. And I think it's very, well, encouraging for us, talking about homology from, oops, it's too loud. In the context of what Chet said before the coffee break, that there are no new parts in the brain in human evolution. So an ape has the same pieces of a brain and the pieces of an endocast, but they have different relative sizes and shapes. And that's why, in principle, they are homologous, right? So I think in principle it's possible to quantify it. And what I briefly want to do is I want to contrast three methods that I think do this pretty well. One of them is our own. And these methods, there are other methods out there that also do well, but I want to focus on these three because they want to do very similar things, and I think from the outside, if you're not really into the mathematics and statistics of this, then they basically do exactly the same. And what I try to convince you is they don't do exactly the same, even if they pretend to, and sometimes these differences might matter, and it's better to be well informed about the possibilities and limitations of the methods that you're using. So I'll talk about endocranial semi-landmarks, which is the approach that Simon and I have been using for quite some time. I'll talk about a paper that was published in 2007, which is a proof of concept paper by Matthias Specht et al., who did this work with a group in Zurich, Marcia Consolidon and Zollikofer, and they published I would say, really interesting approach called spherical mesh parameterization. And I'll talk about a very recent paper by a French group called spatio-temporal deformations, which uses some very clever and complicated mathematics to model endocrinal shape and shape differences in general. So that's what Simon and I have been doing. So what we do is we quantify landmarks on the skull, and we work with curves and semi-landmarks on curves, and we work with semi-landmarks on surfaces. More details to follow later. That's what Matthias Specht has been proposing, the method of spherical mesh parameterization, where you basically start by measuring 7 landmarks that you use to register your endocrinal surfaces, and then they project them out onto a sphere, and then they resample the coordinates on the sphere, and by doing that, they place 163,842 vertices, and that's a subdivision scheme that always comes up with the same number of vertices, and because you've registered the surfaces beforehand, they end up in being in approximately homologous positions. And the most recent method was developed by Stanley Dupont in France, and they're using this in José Pagas group to, in this case, to model the developmental differences between bonobos and chimpanzees, and this is one of their slides. And trust me, you don't want to see the formulas that go into this. It's a bit complicated. So let's talk briefly about the thin plate spline method, and I know that many of you have seen this many times, but it's still worth emphasizing a few things because this is so fundamental and has so many subsequent implications for the way we interpret and analyze our data. So you can compute a thin plate spline deformation grid between 2 forms, only 2, not 3. You only have to pick 2. And so you have a reference shape that you quantify the landmarks. They have to be the same number of points, and they have to be in homologous positions. And so I have a very ugly shape on the left with 6 points, and a very equally ugly shape on the right. And what a thin plate spline deformation grid models is how you would have to end up at the right side. And what the thin-plate spline does is it's an interpolation function that models the space in between where you don't have collected landmark information. And so it's trying to give you a smooth interpolation grid. And instead of warping the vertices of a grid, you could also apply this to a surface. Because a surface ultimately is just, for a computer, it's just a point that is connected by triangles, I mean a set of points, and in addition it has information about which points form a triangle, and that's how you render the surface. So you can warp, using the thin-plate spline algorithm, for example, based on landmarks, you can warp a modern human into a gorilla, and that's what the thin-plate spline based on, I don't know, 20 landmarks or so would do to a modern human cranium, if you also measure gorilla. And with a few hundred landmarks and semi-landmarks, you can pretty much cover the entire range of hominoid cranial shape diversity. So you can see here you have a modern human cranium morphed into a gorilla, a gibbon, a chimpanzee, an australopith, a paranthropus. So I think we're getting pretty close with regards to the shape similarity. And the question is, okay, well, can we do this also for endocrania? And I try to convince you, yes. And so the movie that I just played is actually is a screen capture from a computer game that we programmed in Leipzig for kids that wanted to learn about. Basically, on the open day of the Max Planck Institute, we had this computer game where kids wore a helmet and gloves, and they were basically playing in a tracking system, in a 3D tracking system, and by moving their body, they could change the shape. But what I'm trying to argue is that we don't need to do this only for fun, but this morphing also has some scientific value, because I think this is a way to test whether the homology map that you apply or imply with your measurements makes sense in terms of biology. So what do I mean by that? So here we have two endocasts. One is a chimpanzee on the left-hand side, and on the right-hand side you see a molecule. And if we place 29 points, which is the endocranial landmarks that we work with, and you warp the chimpanzee into the modern human using a thin-plate spline morph, you can see that, okay, I mean, it gets the size right, I would say. But what it doesn't get right, unsurprisingly, are sorry, that is the morph. So I morphed the chimpanzee into the human, and maybe you can see it better, like this. And what it really doesn't do correctly is the morphing of the frontal lobe. Unsurprisingly, as I said, because there are no landmarks there. So I think the spine cannot do magical tricks, so if there are no landmarks, it gives you a shitty answer. So what we can do is we can step up the game, and instead of using 29 points, we can use 307, which I'm doing right now. So if you place many more points on the chimpanzee and many more points on the human cranium, then you can warp the chimpanzee to the human with, I would say, decent accuracy. So this is a view from the side. These are the same surface, but one's the original chimpanzee surface and one's morphed to the modern human. And what you see is that it, I would say, compares favorably to the original human shape. So anyone with paleontological knowledge would recognize this as a form in the back as a human-like shape, even though it's a morph chimpanzee. So what do I mean by this? I think this is an indication that the homology mapping of the thin plate spline works. Gives you a reasonable answer. What doesn't work, of course, is the thin plate spine cannot capture the fine details. You can see here impressions of blood vessels, and of course those, if you don't have landmarks on them, then it's not being morphed accordingly and not correctly. So you also have to be careful about the, I would say, the scale of the information you are interested in. So I think that's a way to test the homology map. Is to apply morphing. And this is another issue that always arises if you compare two forms that are in different coordinate spaces. In this case, you have a modern human, the same orange modern human again, and in blue, this time you have endocranium. And you can see that, okay, they're different in size and they are also different in shape. And how do we compare these two forms? So obviously what we want to do first is we want to bring them into the same coordinate system. And what all these methods do is they compute the center of the shape and they superimpose the centers. So and then what one usually does is you compute the main axis of variation of these surfaces and you align the two main axes. What you see here is that the computer, if you ask the computer to align the main axis of a chimpanzee, of a gibbon and a modern human, it gives you the exact wrong answer because as you see here, maybe it's hard to see, but basically the back of the endocast is registered with the frontal lobe. So the automatic orientation fails. So of course, okay, we can help the computer a little bit. So I manually rotated the endocast and said, okay, try again. So the obvious next step would then be we want to compare shapes, maybe. So we have to standardize size. And one reasonable approach to standardize size is to scale them both to the same volume. So you just divide out the cube root of the volume of each endocast, so they have the same volume, which looks like this. And then, so far so good, that's, I would say, uncontentious and easy. It takes about 2 seconds. But then comes the hard part, and that's how do we superimpose those surfaces in order to compare them. So this is one way. This is the answer that a computer scientist will give you. This is based on surface matching. So you minimize the root mean square between the two surfaces overall, right? And it seems to do a decent job. And then the next thing a computer scientist would do is it would compute, or he or she would compute a distance map between the two surfaces and color code them. And large distances are red, and small distances or no distance is blue. And this is the shape difference between a modern human and a gibbon. And you can see based on this color map that there's a large difference in the frontal lobe, which is true. So it seems to do a decent job, so we got the paper about that and go on, right? The problem is that how these distance maps are drawn and how these colors are distributed over the skull is entirely dependent on the registration. So I told you that this is based on an overall surface fit, which is the standard alignment that every computer program will do for you. But what if we say, okay, maybe it's not just the frontal lobe that's different. Maybe, we know that humans and gibbons have very different relative size of the neocortex, so maybe we want to show that and emphasize that. And we could just move the registration slightly differently and just superimpose them based on the cranial base, which is something I would say a reasonable biologist might want to do, because that would give you a homology mapping that has some roots in biology. So if we have go back again. So I just moved one of the endocastles slightly, and then I recompute the homology map. Yeah, so I have a distance map, and that's what we get. And if we flip back and forth, these pictures are quite different, right? And they are both correct. So there's no way of telling, there is no right or wrong answer here. But I think, would write a very different paper about these two different pictures. Right? So what I'm trying to say, I think, is distance maps only show you part of the information. Distance maps are entirely dependent on your registration, your superposition. That superposition is arbitrary. And I think my summary is distance maps are evil. They might be a very useful tool to visualize some of the aspects, but they should never be your only tool of visualizing shape differences, because they just capture one point of information and they are biased by a totally arbitrary decision about how to match them together. So what do I have to offer instead? So when distance maps are evil, what's good? I think what's better is showing shape differences as deformations. So what I'm showing here is a thin plate spline morph of a gibbon into a human. And you can appreciate that, start it again, yeah. So you can appreciate that, especially if you watch this as a movie, and if you publish this as a sequence of screen captures from that movie, then it's easier to interpret and plain obvious to see where the differences are. You might want to enhance it with a color coding, but always be aware that the color coding is arbitrary. So where does that leave us with our three methods? So I apologize that the font is very small. So how do semi-landmarks and two other different methods compare in these, I would say, tricky aspects of the method? So first of all, you can see here that what I've tried to partition is the data input, then how these points are processed, and how shape-informed differences are compared, the metric of shape-informed differences. So if you want to use semi-landmarks, like Simon and I will do, then for every specimen you need a surface. We use 29 landmarks, we use 60 curved semi-landmarks, you can use more. And for a single specimen, you also need to measure a template of surface points. And then what you do is you map the surface landmarks from that template specimen to all the other specimens using a thin plate spline. You project down onto the surface, then you slide all semi-landmarks to the mean shape. I'll talk about sliding in a bit. And then finally you do your analysis, you register based on all landmarks, and then you scale to centroid size, which is not identical but should be equivalent to scaling them by the cube root of the volume. And then you have shape coordinates and your size separately, and you can do fancy multivariate statistics and beautiful pictures and implement. So what's the deep dark secret of semi-landmarks? I think the deep dark secret is right now this method cannot handle more than around 2,000 semi-landmarks. That's not a mathematical limitation, that's a limitation that comes from how much gigabytes of RAM you can purchase, because you have to invert a huge matrix on the computer, and with the computer that I have now, I can use 2,000 points. So if you want to use 1,000 landmarks, then you have to go look elsewhere or wait until you can afford 64 gigabytes of RAM or so. And I think one of the drawbacks is that the data collection is fairly time consuming. So we're talking about at least 1 hour per specimen, I think, and if you have an incomplete fossil, then it's usually longer. So, so what do you have to do for the spherical mesh approach by Matthias Specht et al.? So for every specimen you need a surface, you need 7 landmarks, and you need to decide what kind of, what they call it, so the subdivisions they're called tessellations, and you have to decide on a tessellation scheme. In this case, they picked a very popular one called the loop tessellation scheme. So what they do is first they register based on these 7 landmarks, then they scale to the same volume, then they make the surfaces to the sphere, and then they remesh the surfaces according to the tessellation scheme. And as a metric they propose the vertex differences, but you could also in principle do Pascal's analysis, principal component analysis, you name it. So what they argue is that we can use these more than 100,000 points as homologous points. And I would say the deep dark secret of this method is that it's entirely dependent on the 7 landmarks that you define initially. So you note that the registration is the very first step. So basically, if the 7 points you pick are not very good, or if they're not particularly homologous, or whatever, then this has this completely determines how the points end up on the netocast subsequently. So basically, even though you have almost 200,000 points per intercast, they are largely driven by the 7 landmarks that you define to register the surfaces. And I think another potential problem is that the application to partially complete specimens, meaning fossils, is not worked out yet. I think there's a way to do this. I think you could do a very similar thing to what Simon and I have been doing, and what I'll talk about in a minute. But anyway, it's not published yet. And finally, I would say based on the labels and how they present the method in the recent paper in the Journal of Human Evolution, the most promising method is the method of spatiotemporal deformations by the French group. And so for every specimen you only need a surface, no points whatsoever. And you start by registering these surfaces, and then you measure the correspondence between the continuous surfaces based on a metric they call currents. And there's some arcane and very detailed and very complicated clever mathematics that go into this measure of currents. I would say one of the problems is that it makes quite a lot of assumptions about the underlying biology of this that might be true but may not be true, and are very hard to, I mean, it's impossible to test based on the data that we have right now. And I would say that the deepest and darkest secret is that it's again dependent on an initial registration step. So again, you start by registering the surfaces, and there are many ways to do this, as I showed you before. But basically, whatever comes next depends on that registration, and that might be, I would say, the biggest problem. One of the potential problems for paleoneurology is that it's unclear, at least to me, how one would extend this to include partially complete specimens. Because right now, what they do is they take the surfaces and they compute the surfaces as a whole. right? I mean, like the entire surface of one intercast to the entire surface of the other intercast. And how you would do that if one of the intercasts is incomplete has not been worked out yet, and I don't think anyone can, but I'm happy to be proven wrong here. So you see that these methods do very similar things, but they do them slightly differently, and depending on what you want to do, one method might be better for a particular task than others. So, one question that I always get, so I want to address this in this forum, is that the semi-landmarks that we use, they're called sliding semi-landmarks, and they're called sliding semi-landmarks for a reason, because they're not equidistant points. We start by using equidistant points on curves, and then we allow them to slide. And the question always is, do we really have to slide them? Does it really matter? So what I try to show you is one example where I think it really matters. So these are data from research sections of T1 with weighted MRI scans, so normal clinical MRIs. These are of healthy individuals. It's actually a cross-sectional series, but it doesn't really matter. And what we measured is the outline of the corpus callosum, the structure that connects the two hemispheres of the brain. And what you might see here on the left is how we digitized the corpora callosum. And you notice that there's only one little dot that has a slightly yellow tint on the video projection, and that's the only real landmark we used in this study. And all the other points are equidistant landmarks around the corpus callosum, which I think is a decent way of quantifying the shape of that curve. And what we do is we don't analyze the equidistant points, but we allow these points to slide along the curve, preserving the original shape until this thin plate spine deformation grid on the left-hand side between each form and the mean shape is as smooth as possible. So we allow these points to move because ultimately what one has to realize is that equidistance is a very convenient way of distributing these points, but it's an arbitrary one. And it does not go well, this arbitrary space does not go well with how the spline deformation visualizes shape differences. And that's why we basically optimize the position of the semi-landmarks so we can interpret the spline space, so we get the smoothest possible spline. That's the idea. And if you want to read more about this, I can give you PDFs of papers later. So I think what I try to show you now is why they really have to slide. So this is a principal component analysis, typical multivariate analysis of the shaped trends in a data set. And what you see is that there's a strong first principal component. So this is the x-axis, and these are the visualizations. These are the shape changes that are associated with these axes. And these are the equidistant points. So now we allow the points to slide. And then what I did is I projected the slit configurations into the principal component space and see where they fall. And you can see that by sliding, the red ones are the same forms after sliding. They completely change the entire picture. So the first principal component, the dominant mode of variation in the entire dataset, was entirely driven by the arbitrary space semi-landmarks. And if you optimize the position of the semi-landmarks, you get a completely different principal component. So that's the principal component of the slit coordinates. And you see that the pattern of variation looks very different, and also the visualization of the shape change looks very different. I zoom in for a second. So this is the visualization on the left-hand side of the first principal component before sliding, the equidistant, which you would interpret as, okay, there's something really weird going on here. So you have, because the spline bends here, right? So you seem to have a very localized shape signal, whereas here you have a very general, very smooth trend with this part of the corpus callosum moving slightly upward. This is like the opposite paper you would write about this finding. Right? So in these cases, in many other cases, sliding does matter. And the problem is, unless you haven't slit them, you can't tell what the difference would have been. So I would strongly suggest not to rely on equidistant spaced curves, especially or points on surfaces, especially if you have an extreme case like this where the points can move around the structure.
Sorry, Philip, but in this case, how can I know what of the two models is closer to the real natural signal of the morphological variation?
The thing is, the thin-plane spline doesn't try to model the biology, and what you're doing is you're not changing the shape of the curve. Like each specimen, by sliding, you're leaving the shape of the corpus callosum the same. And so you can't show here, but basically I could show here. If you work in the middle, okay, there's a margin of error because actually they slide on tangents, but you could project them to really preserve the shape precisely. But the shape itself doesn't change at all. But what changes is the thin plate spline that shows you what the shape differences between two forms is. And the argument, our argument is if you visualize by thin plate splines, you have to slide. And you have to slide to minimize bending energy.
Well, I was not worried about the visualization.
So what this tells you is that this axis is not a biological one. It's an arbitrary one that depends on your decision to sample the curve. If you, I think I took 90 points, 90 semi marks. If you took 20 or if you took 200, the principal component would be totally different. And if you slide, then taking 20, taking 30, taking 200 will look the same. That's, I think, the core of the argument. That the spacing of the points has nothing to do with the shape. It's an arbitrary decision, as it would be the registration. The sliding gets rid of that arbitrariness of the spacing.
And it is published this, this different sampling with different number of landmarks using the two approaches. This is your personal experience, or it is published in this paper?
I did not include the figure. Maybe I should have. I'll publish it. It's easy to show. Of course, you have to sample the curve somehow. So you can't just take 5 landmarks because that will not adequately represent the complexity of the shape. But I think we're converging on the true shape here. So I think what one would have to show is, okay, 50, 100, and 200 give the same result after sliding, but they don't give the same result with equidistance.
Because, for example, for a simpler profile, I did the same job with a cranial bolt profile, and I can remember that 5, 10, 50, equally spaced without sliding, no difference at all.
I think that I agree with you. If you have this is kind of an extreme case because the surface or the curve turns around itself. But actually, we have the same in the endocast, right? Because you know, if you have semi-landmarks on the frontal and they can be on the orbital floor, it's basically a 3D corpus callosum. It's the brain is the 3D version of this.
So it may be a matter of complexity of the geometry.
Yeah. And the thing is, usually, I have to admit, I was totally shocked when I computed this figure, because I actually wanted to show, initially I wanted to show something else. I wanted to show, okay, it matters for the thin plate spline visualization, for basically for this, but it doesn't matter for the pattern in the principal component analysis, which is what you were talking about before, which is my experience with other data, that usually the first few principal components look the same, more or less the same. But in this case, it didn't. And
I think that this should be published. You know, we have just this year we have published 2 papers on the corpus callosum, and in both papers we use slide landmarks, and in the methods we write we use slide landmarks because Bookstein and Gunz tell we have to do it.
Yeah, no,
so a publication
is not enough.
So a publication will be very interesting.
Yeah, it's true. It's a good suggestion to publish this. I recommend you as a referee. So I think this is the part that I would like really much. It's about how to quantify symmetry, and it seems to me that that we're doing very similar things. Only we're doing it with endocasts, because there are a few, I would say, proposed methods out there in the literature, in the anthropological literature, how to look at endocranial symmetries. And what's common to them is that they define a midplane, and they use different ways of measuring the distance of points or structures on the left side and the right side of the brain from that perfect midplane. The problem is that, first of all, there is no perfect midplane, because that's the very nature of the brain asymmetry, that the midline is curved. And second of all, not even Ralph Holloway and Tim Falk can agree, okay, they can't agree on anything, but even the mid-sagittal plane of the town child is a place to fight. And I don't think that it speaks to the character in this particular case, because sometimes it's hard to define a plane, especially if you rely only on external landmarks, or even internal landmarks, because they don't have to match up with the brain asymmetry, right? Because this is a totally different process. Basically, where brachioma is on a skull is determined by different bone remodeling processes than where the midline of the brain is, and they usually are highly correlated, but there need not be anyone who's looked at an archaeological sample or any anatomical collection knows that there are sometimes very asymmetrical skulls on the outside, but they look perfectly normal on the endocast. So I think it would be very nice if we had a method that would not rely entirely on outside landmarks. And it turns out there is a method. And the method uses the Procrustes, the geometric morphometrics approach to symmetry. That's been published by Kanti Martia and Fred Bookstein in 2000, I think, and has been applied many times now, and Chris Klingberg also uses the same method, which I think is what I is referring to. And the idea is this. So I'm visualizing this using just the lower part of the brain, of the endocast, sorry. So this is the cranial base, right? So here we have the posterior cranial fossa, the foramen magnum, and this is the midline, right? So we're looking at this from above. And so how you start is you measure points and you define which points are pairs. That's basically the data input that the algorithm needs. And then you reflect along any axis. Can be anything. You could flip it the other way around, doesn't matter. And then you relabel the points. So first you reflect, then you relabel, and what do you mean by relabeling? You switch the labels of left and right paired points. And then you superimpose them, and you can see this is the blue shape is the original shape, and the orange shape is the superimposed reflected relabeled shape. And if you average these two configurations, you end up with a perfectly symmetrical shape. And of course, the difference between this perfectly symmetrical shape and the original shape gives you the ideal measure of asymmetry. And the beauty is that you can also partition this into directional asymmetry if you have a sample, and what remains after correcting for directional asymmetry, you're left with fluctuating asymmetry. And there are many applications in developmental studies where these two measures are highly relevant. And I'm not going to dive into the details of this, but all I want to show is one analysis that we just did based on chimpanzee and modern human crania, endocrania, with that method. And here we are basically analyzing the residuals of the asymmetry. And these are the principal components of these residuals. Modern humans are in blue and chimpanzees are in red. And you can see that they're not perfectly identical, but the pattern is entirely the same. So the main mode of variation, and we can actually visualize with the first principal component, which is, I think, a very common picture in the study of asymmetry. You can see the petalia in the frontal and occipital lobe, and this is the same between chimpanzees and humans. There are differences, especially in directional asymmetry, but these main modes of variation are the same, which we knew before this slide, but I think with these methods we can now confirm this with, I think, more statistical rigor. And so the advantage of this is that we have pair points that yield the symmetry axis, and it does not rely on external data, and the midline here, and it provides you a direct measure of directional and fluctuating asymmetry. So the big thing for paleoneurology is then, okay, can this be applied to fossils? And my honest answer is, well, nothing's stopping you. I mean, the mathematics don't shy away from fossils, but I would not recommend it, because actually what we usually do is use the very same method of symmetrizing to get rid of plastic deformations. And I think most of the fossils that we, so I think, is this actually moving? Yeah, so this is the Arago skull, and I'm symmetrizing it using reflective relabeling to get rid of that Teflon shear. Of course, once you have symmetrized it, it makes no sense to use the same method to study its asymmetry, because now it's perfectly symmetrical, right? So all I'm saying is one can apply this to fossils in principle, but one should be very careful, and I think one would have to have very good reasons to argue that this is not tephonomically deformed. And if that's not possible, then one should use this approach or any approach for that method to study asymmetry in fossils. On this broad scale, I would say. So, and the final thing I want to mention, but I see I'm a bit out of time, is that we're talking about uncertainty, right? And the uncertainty comes from having incomplete fossils that you can make the computer puzzle games on a computer, but you still end up with parts that are incomplete and that cannot be reconstructed by neural imaging. And that is an inherent uncertainty. And I briefly want to take you through the steps that we have that we're using to get closer to a complete shape and to deal with that uncertainty. So I just want to show how we deal with the town child. So you always start by exploiting the symmetry information. So you mirror image across either a mid-sectional plane, or you use the reflective labeling to exploit the mirror symmetry, because that is, I would say, the least dangerous approach to fossil reconstruction, because the assumptions that we make are very easily argued. You can say, okay, this is probably also a bilaterally symmetrical organism. And then what we do is we use the thin plate spline deformation to estimate the missing parts based on reference crania. So here, for example, we estimate the outer shell of the neurocranium based on a human. And of course, you can use another approach. You can use a chimpanzee. And these are the two resulting reconstructions, and they're not the same, but I challenge you to find the differences. And the differences, of course, reflect the differences between the two reference crania. So we thought, why stop at 2? Why not do 200? And the idea then is to create multiple reconstructions, as many as you can, and look for consistency among these reconstructions. For example, what we did with undercrania and endocrania of Australopithecus africanus to deal with that uncertainty is we've created multiple reconstructions using humans and chimpanzees, but mostly chimpanzees and humans, just to test, I would say, the potential pathologies of that method. And so for every specimen you end up with a distribution of points, and then you, for example, one way to record this data is you take the mode of the variation, the most common value of these multiple reconstructions, and you also take the standard deviation. So there are many measures that you can take from a distribution to tell you something about the accuracy and missing data uncertainty. This is what happens if we look at shapes, and here what we see is that so these are humans and chimpanzees, and these are principal components, and you see that these different reconstructions they cluster together pretty tightly, and that I think gives you a good sense of how reliable these reconstructions are. And of course, specimens that are not as well preserved as STS60, they have a wider range of shape and also endocrine problem estimates. And what we also did is we used simulations to try to test how accurate these methods really work, and we simulated the case of having a very small sample within modern humans and within chimpanzees, and what we found is that the biggest problem comes from the small sample sizes and not the estimation uncertainties. So having a small fossil sample, that's the key issue, and of course one way to overcome this, or to at least move towards a better solution, is to include reconstructions in your estimates, because of course if you ideally you will only have complete specimens, but with Australopithecus africanus you have with STS5 and then have a sample size of 1. And in the last final minutes, I briefly briefly want to mention something, and I can go through this very quickly because Simon is going to pick that up tomorrow. I briefly want to talk about the paper, or the series of papers, one should say, that Jack already mentioned about the developmental differences in endocrine ontogeny between modern humans and Neanderthals. And I only want to focus on two methodological aspects. So I'll skip all of this. And as you know, of course, modern humans and Neanderthals look differently from the outside and also from the inside. And Emiliano has been preaching for years that one should not only focus on the frontal but also on the parietal lobe. And it's unclear when these ontogenetic differences, of these group differences emerged during ontogeny. So we looked at a cross-sectional sample of modern humans and chimpanzees, and what we found is that the first year of life is different between chimpanzees and humans. So during the first year of life, and there you see me using a distance map, our heads become globular, right? And the question then becomes, okay, do Neanderthals also have a globularization phase? Are Neanderthals developing the same way as modern humans do? Or do they develop like chimpanzees do or other primates do? And what we try to do is, and I only want to talk about the methods aspect and the uncertainty aspect of this. What we try to do is come up with smart ways of addressing that difficult question, because we have a decent fossil record for Neanderthals. It's the best subadult fossil record of any fossil species, but still we're talking about very few specimens, and they are very, very fragmentary, especially the newborns. And the other thing is we also have to deal with the uncertainty of these growth trajectories. So just to put that in perspective, because usually I only show pictures of skulls, but here I can show what these pictures relate to using my own son. Because over the last half year, I've been conducting an intimate developmental study with a sample size of 1. So basically, this is a few days after birth. These crania look extremely elongated, and that's what he looks like now. So he's almost done with his globularization phase. It's really the first year of life we're talking about here. And as you know, these first years are very important for cognitive and development and motor skills. So he's a very different animal now than he was 9 months ago. So what I want to emphasize though is how can we answer the question whether Neanderthals and modern humans developed in the same way? And the question I think that we have to, or the thing that we have to rule out, did they develop their brains exactly the same way? Because if the answer is no, then I think it follows, well, then they developed them differently. So, and the limitation is we have a small subadult fossil sample of fragmentary specimens. And so what we're doing is we take the adult Neanderthals, which we have a decent idea about because the sample sizes are decent and the fossils are much more complete. And we say, okay, if Neanderthals and modern humans developed exactly the same way, then we can just take the modern human ontogeny, reverse it, and predict the shape for a Neanderthal baby. And you can see that the fuzziness that comes from bootstrapping. So I also tried to include the estimates of the uncertainty. So if the developmental patterns are exactly the same, then a Neanderthal baby would have to look somehow like this. if Neanderthals don't have this globularization phase, then a Neanderthal baby would have to plot here. And then we can create many, many, many reconstructions of our subadult Neanderthals, and we can plot them here. And many reconstructions form a distribution, and no matter how we reconstruct these Neanderthals, no baby, and we have Lemus-J2 and Mesmeskaya, which are newborns or perinatal specimens, and no matter how you reconstruct them, they do not fall in prediction A. And they all fall within or close to prediction B. And from that alone, we can basically rule out that Neanderthals and modern humans developed exactly the same. And what we can also do is we can predict what a Neanderthal baby would have to look like if it would undergo a globularization phase, and it would have to look like a real freak, right? I mean, because Neanderthal is very elongated as an adult, so it would have to be extremely elongated as a baby and not have an occipital, which is unlikely in the first place, and there's no way you can reconstruct Lemus-J or Mesmeskaya to look like this. And finally, what I want to show is, so how robust is this finding? And there's one other way to test this, and to conclude that this is a very robust finding. And what we did is, we did a developmental simulation and said, okay, what's the weakest part of the argument? The weakest part of the argument is the reconstructions of the subadult Neanderthals, because there's a lot of interpretation, a lot of expertise knowledge that goes into this, but I might be completely wrong. And so, and Marcia and Christoph from Zurich might be completely wrong too. So, can we also show this without using any fossil kits at all? And we can, because if we just do a developmental simulation and look what a modern human baby would look like if it developed without the globularization phase, then they plot not identical, but very close to the actual Neanderthal adults. There are some differences between this prediction and the actual Neanderthal mean shape, and these differences are actually consistent with what we find in terms of shape differences between Mesmeskaya, Lemus-J, and modern human babies. So there are some differences that are already present at the time of birth, but everything else develops subsequently, and only modern humans have this unique pattern of independent development, whatever it might mean for in shape. And of course, we will never have beautiful pictures like this. This is a diffusion cancer imaging picture of a human brain, where you can see the large-scale connections in an adult human. This data we will never have. But we have the comparative data from living apes, and we can also use the timing of these differences and the nature of these differences to make some inferences about cognition and behavior, and that's what CENIEH is going to talk about tomorrow, and that's the end of my talk. Thank you.